We're gonna learn about some heredity.
Welcome to another virtual biology lecture. Today we're going to be discussing genetics part 1. Hope you guys enjoy and don't forget to take good notes. Today's virtual lecture's objective is to become familiar with the following assessment standard from reporting category 2, which is going to be tested in the STAR Biology EOC. At the end of the lecture, you should be able to predict possible outcomes of various genetic combinations, such as a monohybrid cross and a dihybrid cross. Genetics is the study of how traits are passed down from one generation to the next. But in order for us to understand how these traits are passed down, we must first understand a little bit more about genes. Let's first review what we already know about genetics. First, let's start with chromosomes. We know that chromosomes are made up of DNA and that they're found in the nucleus. We also know that chromosomes have a lot of sections in their DNA. Well, we call these sections genes, and these sections are used to control specific traits. Now, a chromosome has many genes, which all control different traits. So how is it that these genes control specific traits? Our genes give the information regarding to observable characteristics such as hair color, eye color, skin color, tall or short, which are all referred to as traits. Now remember that there are a variation of the same genes. Basically, different forms of the same gene are called alleles, which can lead to different traits. Alleles are represented by letters. For instance, in a gene related to albinoism, the alleles could be a big A and a little a. An individual must have two alleles present in order to express the trait. You might be thinking, well, why two? One must come from the dad and the other from the mom. Remember, we talked about homologous chromosome pairs. Each pair consists of one chromosome from the mom and one from the dad. These chromosomes are said to be homologous because they are the chromosomes that carry the same type of genes. Basically, they're talking about the same information. However, this doesn't mean that they are expressing these genes in the same way. For instance, in this example, you will have two homologous chromosomes that carry the gene for albinoism. The mom's chromosome has a lowercase letter which represents a recessive allele which expresses albinoism, and the father's chromosome has an uppercase letter which represents a dominant allele which expresses normal pigmentation. Together they will provide the individual with a capital A lowercase a trait, which is known as a heterozygous trait in which the dominant allele is seen. We will get into more details about this and these terms later on in this lecture. So now that you're starting to see the big picture of our previous lectures, let's now take a closer look at the specifics of genetics, starting with its history, which revolves around Mr. Gregor Mendel's research, who is often referred to as the father of genetics. He was a monk who conducted a series of experiments with pea plants grown in his garden. Mendel is credited for the development of the three basic laws of inheritance, known as the law of dominance and recessiveness, the principle of segregation, and the principle of independent assortment. Now, we're going to be discussing each of these in today's lecture. Overall, he's responsible for demonstrating that genes carry characteristics that are inherited. Let's take a brief look at Mendel's research. As we mentioned, his experiments used pea plants. In order to better understand his experimental procedures, it's important to note this plant's method of reproduction. Pea plants are normally self-pollinating. Since the male and the female's reproductive structures are enclosed inside the flower, the pistil produces egg cells, and the stamen produces pollen, which contains sperm cells. Now, the sperm cells of the flower will fertilize the egg of the same flower. The resulting offspring will have the same characteristics as the parent plant. Mendel identified these plants as true breeding, meaning that if the plants self-pollinated, they would produce offspring that were identical to themselves. Well, Mendel came up with an idea that changed science. He wondered what would happen if he joined an egg and a sperm from different plants. He developed an experiment, but in order to run his experiment, he needed to make sure that the plants were unable to self-pollinate. In order to prevent this from happening, he cut away the stamens and dusted the remaining female structure with pollen from a different plant. Now, this is known as cross-pollination, which gave Mendel the opportunity to experiment with different plant characteristics through crossbreeding. But before we continue to look at Mendel's experimental findings, there are a couple of key words that you need to be familiar with. For instance, Mendel referred to the initial organisms that were bred as the parental generation, which is often abbreviated as P generation. Then the offspring that came from this breeding was referred to as the first generation, and it's abbreviated as F1 generation. Then the generation that followed would be referred to as the second generation, and so on. Now, there is one other term that you need to be familiar with, and that is a hybrid. Now, this term is used to describe offspring of parents with different traits. Let's take a look at one of his early experiments in which he decided to cross true breeding tall plants with true breeding dwarf plants. But what does this mean? Well, this means that he obtained a tall pea plant that was known to self-pollinate and a dwarf pea plant, in other words, a short one, that was also known to self-pollinate. But before they could each self-pollinate, he crossbred them, like we mentioned before, in which he cut the stamens and dusted the remaining female structures with pollen from the other plant. Mendel noticed that the F1 generation of hybrids were all tall. Notice we refer to this generation of offspring as hybrids because they were a cross between two parents with different characteristics. Mendel didn't understand why all of the offspring were tall and none had the dwarf trait. He began to think that maybe the dwarf trait had been lost. For the purposes of describing a cross or a breeding like this, we tend to write it out as tall for the trait tested, and the multiplication symbol means that it was crossed with a dwarf, the other trait, which yields a tall offspring. Now, inheritance is determined by genes that are passed down from one generation to the next. Each of the traits that Mendel observed in the pea plants was controlled by one gene that occurred in different forms or was expressed differently. For example, the gene for the height of pea plants had two different forms, tall and dwarf. The different forms of a gene are called alleles, which are represented by letters. Note that when two parents that express a single gene differently are crossed, we call this a monohybrid cross, which is what Mendel did in this experiment when he crossed a tall and a dwarf plant. Some alleles are dominant and others are recessive. An allele that covers up or masks another allele is known as a dominant allele, which covers up the recessive allele. So what does this mean? Well, let's say that a dominant allele is present in an offspring. Then that means that the dominant trait will show up in the offspring. We represent this dominant allele with an uppercase letter. Now, a recessive trait, on the other hand, only shows up in the offspring if the dominant allele is not present. Instead, two recessive alleles would have to be present, and they are represented by a lowercase letter. Now, upon obtaining these results in which all of the F1 generation was tall hybrids, Mendel started to ask himself another question. He wondered whether or not the dwarf trait had completely disappeared, or was it still present in the F1 offspring, as perhaps a recessive allele. Well, he tried another experiment to test out whether or not this was true. He ended up allowing all of the hybrid offspring from the first generation to self-pollinate and then recorded his findings. This is where he realized that the recessive trait was not lost. It was simply being hidden by the dominant trait. The second generation of offspring included 3/4 of the pea plant's population to be tall, and 1/4 of that population were dwarf plants. Then, when the second generation was produced, that dwarf trait had been passed down and was allowed to reappear. But why? Well, this is where Mendel thought about it and said, the organisms must have two alleles for every trait, which are inherited, one from each parent. Basically, if the offspring receives a dominant allele from one parent, that dominant trait is going to appear in the offspring. However, recessive traits only show up in the offspring if the offspring receives two recessive alleles, one from each parent, which is what we saw in the second generation, since all of those in the first generation were carriers. So you might be thinking, how is it that if each individual has two alleles for a trait, then how do parents only pass one allele to the offspring? Remember, each individual has two alleles, basically two letters for every trait. In other words, one allele comes from each parent. So let's think back at meiosis. We said that this is the cell division used to produce gametes, which are sex cells such as egg or sperm. Remember, this is where homologous chromosomes are paired up and then split up, and each chromosome has different alleles expressing the traits represented by the genes found on those chromosomes. Well, if we just ignored those homologous chromosomes in the background and only focused on one specific gene controlling a specific trait, we would see the allele found on each chromosome giving us 2 alleles for that specific trait. Remember that the capital letter T represents a dominant allele, and the lowercase t represents a recessive allele. Remember, each chromosome after interphase will have 2 chromatids per chromosome because DNA has been replicated. Now this means that you're now going to have 2 of the same alleles for each chromosome, giving you the tetrads. In the first round of cell division, known as meiosis 1, the 2 homologous chromosomes split up, leaving you with 2 of the same alleles, same chromosome, on one cell, and the other 2 on the other cell. Then the second round of cell division, known as meiosis 2, comes around, and now you split up those 2 alleles in each cell, giving you a total of 4 gametes or 4 sex cells, each with 1 allele for each trait. Now, Mendel's principle of segregation says that every individual carries 2 alleles for each trait, and that these alleles will separate or segregate during the formation of the egg and sperm cells. Basically, a parent passes 1 allele for each gene to their offspring. Then based on probability, which is what we're going to be discussing in this lecture, a sex cell will combine with another, making a diploid cell called a zygote, which will once again have a homologous pair of chromosomes carrying these genes with 2 alleles once again. Therefore, an offspring will inherit 2 alleles for a trait, 1 allele from each parent. The combination of alleles received may either be homozygous or heterozygous. Homozygous means that the 2 alleles are the same, either both dominant, 2 capital T's, or both recessive, 2 lowercase t's. Heterozygous, on the other hand, means that the 2 alleles are different, a capital T and a lowercase t. In other words, 1 dominant and 1 recessive. Now, we describe these combinations as genotypes, which are the genetic makeup of an organism. Now, what about phenotypes? Well, phenotypes, on the other hand, these are the physical characteristics of an organism, basically what that organism looks like. Let's look at Mendel peas plants as an example again. Remember that the tall allele was dominant over the dwarf allele, which is why we're going to say that the capital T is the dominant allele and the lowercase t is the recessive allele. Basically, we know that there are 3 combinations of alleles, in other words, 3 different genotypes: homozygous dominant, homozygous recessive, and heterozygous. As for the phenotype, we know that whenever a dominant allele is present, the dominant trait will be expressed, which means that both homozygous dominant and heterozygous will look tall, and the only dwarf will be the homozygous recessive. So now that we know what the different allele combinations could be, how could we determine what the probability would be to obtain those traits among the offspring? In other words, what are the chances that that offspring will look a certain way? Let's say tall or dwarf. Well, we could use a Punnett square to determine the percent probability in a monohybrid cross. Remember that we said that a monohybrid was a cross between parents that differ in 1 trait. Basically, we are only taking 1 trait into account. So first, we draw our Punnett square, which is a diagram showing the allele combinations that might result from a genetic cross between 2 parents with a different trait. The Punnett square is made up of 4 squares, each representing the possible offspring's genotype. Now that we've drawn our Punnett square, let's label it. For the purposes of this example, we're going to use Mendel's first cross between homozygous dominant tall pea plants and a homozygous recessive dwarf pea plant. The genotype for this first parent was capital T, so we're going to write its alleles across the top of the squares. Then the genotype of the second parent was lowercase t, so we're going to write these alleles along the left side of the square. Now we need to determine all of the possible gene combinations of the offspring which will be placed inside of these squares. Remember that the capital letter represents a dominant allele and the lowercase letter represents a recessive allele. Notice that the phenotype for all of these offspring would be tall because a dominant allele is present in all of them, which will cause the recessive allele to be hidden. We would describe these findings in a T-chart like this, in which the genotype of 4 out of 4 offspring were heterozygous, and as for the phenotype, 4 out of 4 were tall. Basically, we could say 100% of the offspring were all heterozygous tall. Now let's take a look at what Mendel's second experimental findings would have looked like if he allowed the first generation to self-pollinate, meaning the parents would both be heterozygous. Okay, so we write these findings in a T-chart. We would see that the genotypes for the cross would be 1 out of 4 homozygous dominant, 2 out of 4 heterozygous, 1 out of 4 homozygous recessive. And as for the phenotypes, we would know that both homozygous dominant and heterozygous would be tall, so that means that 3 out of 4 would all be tall, and only 1 out of 4 would be dwarf. This means that 25% would have a genotype of homozygous dominant, 50% would have a genotype of heterozygous, and 25% would have a genotype of homozygous recessive, meaning that as far as the phenotypes go, 75% of the offspring would all be tall, and only 25% of the offspring would be dwarfs. As Mendel continued his research on various pea plant traits, he began to wonder about the effect that one pair of alleles may have on the segregation of a different pair of alleles during meiosis. In other words, do genes affect one another? For instance, does the gene that determines the shape of a pea, round or wrinkled, have any effect on the gene that determines the seed color? Now, this is where Mendel began his dihybrid crosses experiments, in which he tested two different genes such as pea shape and color found among parent pea plants and identified the resulting offspring. He concluded that the alleles of genes on non-homologous chromosomes assort independently during meiosis when gametes are formed. For instance, the first homologous pair consists of the gene responsible for determining pea shape, which is labeled with the letter R. And as for the second homologous pair, it consists of the gene responsible for determining seed color, which is labeled with the letter Y. Remember that based on the law of segregation, each homologous pairs will segregate into 4 different gametes. Basically, only one R per gamete and only one Y per gamete. Now, the law of independent assortment simply states that the non-homologous chromosomes, for instance, a chromosome with the R allele and the chromosome with the Y allele, will separate into 4 different gametes independently from each other. Basically, if the first gamete obtained a dominant Y, it doesn't mean that it can only obtain a dominant R. It can actually assort independently, meaning various combinations can be made. Okay, so let's take a look at a dihybrid cross, in which we're going to be focusing on fur type and color for these dogs. Note that the black fur is dominant over yellow fur, and the straight fur is dominant over curly. Now let's say that a dog with black curly fur is crossed with a dog with yellow straight fur. Then allow the first generation to crossbreed. What percentage of the second generation of puppies would have yellow straight fur? Now, in order for us to set up our Punnett square, we first need to determine what the parental gamete combinations would look like. And we do this by using the FOIL method, in which we look at the first parent and determine its combination starting with the first, outside, inside, and last. Now we're going to place all of these 4 combinations on the top of the Punnett square. Now as for the second, we start again with first, outside, inside, and last. And we will place these 4 combinations on the left side of the Punnett square. This will give us the following possible offspring combinations. Notice that 4 will have a homozygous recessive genotype, meaning that they will express the yellow fur phenotype, but only 3 of these will have straight fur. Thus, 3 out of 16 will have yellow straight fur, which means that there is less than 20% chance that a puppy from the second generation will have yellow straight fur. Looking at the big picture here, we can see that Mendel's 3 major principles form the basis of modern genetics, which state that the inheritance of traits is determined by genes, and that these genes are passed down from parent to offspring. Overall, each gene must have 2 or more forms called alleles, which some are dominant while others are recessive. Now, it's important to note that each parent has 2 alleles for a specific trait, and they inherited these from their parents, but they will only pass down 1 allele to their offspring when the alleles are segregated into gametes. Now remember that the alleles for one trait will segregate independently from the alleles for another trait. Okay, now that we've gone over the 3 laws of inheritance in this Genetics Part 1 video, you should be familiar with the following assessment standard from Reporting Category 2. This concludes our virtual biology lecture on genetics part 1. I hope you all took great notes and wrote down any questions or concerns that you may have so that we can discuss in class. Well, that's it. Have a great one.
We're gonna learn about some heredity.